welcome everyone to the Brain Space Initiative. Today it is my honor to introduce Christos Stavitsikos. He's currently the Wallace T. Miller Senior Professor of Radiology at UPenn, and he's directing a center called AI2D for AI and Data Science for Integrated Diagnostics. He's also involved in this sort of large effort, AI for AD. And so he's going to be touching on, I think, a really interesting topic, which is how do you deal with heterogeneity, when you're studying neuroimaging data. And so I'm really looking forward to your talk and really glad you could join us today. So I'll let you go ahead and take it from here.
Thank you so much, Vince. Great to join you guys and great to see you again. Before I get to the issue of heterogeneity, let me just do a quick kind of nostalgic trip back 20 years when in the old days we were trying to use machine learning in neuroimaging, but Vince and I and a few others who were living in this field, but people used to say it's a black box, it's not going to work. Of course, now everybody is excited about the AI. Back then, we were trying to, do mundane things such as classify whether. Let me try to get my laser pointer. You see my laser pointer?
Yeah.
Yeah. To classify whether an MRI belongs to a man or a woman, which is a very mundane task. Then we were doing some more interesting things, such as trying to find an imaging signature of schizophrenia from MRI. So in 2007, there was some work that Yongfang, who is now on our faculty here, but he was postdoc back then. It was one of the papers that showed that we can actually find an imaging signature of Alzheimer's disease that you can detect at the individual level. And of course, there have been a lot of group analysis that show differences between patients and controls, but to be able to identify a signature in an individual, it was not really possible until these machine learning tools came along and allowed us to form multivariate patterns. What was most interesting back then is that you could actually predict whether someone with a mild cognitive impairment at baseline would progress to Alzheimer's disease or would remain stable. And again, that was based by combining simply an MRI scan, a clinical test, and ApoE genotype. And what was even more interesting, I think, it was that you can actually start detecting these subtle patterns of neurodegeneration and MRI in people who are cognitively normal. They don't have any symptoms, and they remain cognitively normal for many years. But some of them progress, and the people who progressed are here on the right. This is results from the Baltimore longitudinal study of aging back then in 2009, and which the y-axis shows the rate of change of this pattern. So in other words, as the machine learning model is applied to longitudinal data, it sees an increasing expression of this pattern of atrophy in people who eventually progress to dementia versus people who remain stable. Excuse me. So we were playing with these things for a while and then in different contexts, but at some point it became clear that, number one, you need a lot of data. So machine learning, especially as the models evolved into deep learning in 2012 to 13 or so, it became very clear that you need a lot of data. So that's one of the problems that we had, that you were working with 500 data sets and it is all cool, but when you try to apply it to a new data set, it was always challenging to have a model that is reproducible, sufficiently reproducible and robust. You need to have a large data set and, most importantly, diverse data set, so that you can actually see a lot of different patterns and be able to build a robust model. The second issue is heterogeneity. And now this will be the main topic of much of my talk today. That when we're trying to talk about an imaging signature, say of schizophrenia or Alzheimer's disease, there is not a single signature. Or cancer. These are very heterogeneous diseases and disorders. And talking about a single signature, it's really not correct. You really need to tease out the heterogeneity, and potentially some of these diseases might have completely different neurobiological underpinnings. So in the past 8 years or so, we've started to spend a lot of effort on what we call, what belongs to the family of weakly supervised methods. So it's not clustering, which is unsupervised, but it's not completely supervised method where you're telling it these are examples of patients with the Alzheimer's disease. This is examples of normal controls. Try to train a classifier to differentiate between the two. So to put data together, we've formed a number of consortia. We've been very fortunate because people are willing to collaborate and share data either with, in pairwise agreements or commonly, openly. A lot of these data sets are now available. But with the iStaging Consortium was the first consortium we put together. It still goes on to try to understand brain aging and try to put together data from a variety of studies. I think it's now over 26 studies or so, and over 85,000 brain MRI scans from different populations. Some of them are normative data of midlife or aging, and some of them come from databases like ADNI or UK Biobank, and some of them are specific studies to diabetes, hypertension, and so forth, sociodemographics and so forth. So we try to learn as much as we could, and obviously, as you all know, much of the work now in deep learning revolves around getting a lot of data to learn from, because you really need to sample the diversity of the underlying distributions to be able to derive robust models. This is the AI for AD consortium that Vince mentioned before. This is 5 of us are the lead PIs, and there's a bunch of other people who are also co-PIs who we're putting together data, MRI, whole genome sequencing, and clinical data from a number of centers around the country, particularly AD clinics, to be able to offer a data set, hopefully over 50,000 people, with characterization of imaging, genomics, and cognition, so that people can start mining the data and try to find associations between imaging and genomics and so forth. We had a consortium, the Phenom Consortium in psychosis, where we put together data from a number of studies of people looking at primarily schizophrenia. We always look for collaborators who are interested in sharing their data, and these consortia are actually dynamically expanding, but they are platforms for people to share their data if you want, for us to train and test machine learning tools on. So the moment you start to do that, as I'm sure most of you know, is that you start to run across variations across scanners and protocols. Different studies use different MRI protocols, and it's not straightforward to combine the data. So one of the areas that we spend some emphasis on is statistical harmonization across studies. And my colleague here, Hao-Chang Xu from Biostatistics, has been, along with Taki Shinohara, also from Biostatistics, have been leading some of these efforts of applying a family of methods that is based on combat, which I'll describe in a second, into neuroimaging data. Combat is a technique that was developed. It's an empirical-based estimation method that was developed for genomic data, because people doing genomics also have the same issues of different assay effects. As you apply a different assay, you might get slight shifts in the signals that might actually shift the distributions. So you cannot really combine data before you do something to harmonize. So we took these methods, or Hao-Chang and Taki and others took these methods initially, applied them to neuroimaging, and we further built upon that by adding some generalized additive models to allow for nonlinear effects of age. So we can actually effectively stitch all the studies together if you want. And this shows you age strengths of different brain regions as a function of age. For different colors correspond to different regions. This is fMRI signals before and after harmonization. So that's a very important step before you start putting all the data together and then training a machine learning tool to do that, to do something, such as develop identification of disease or so. Another family of methods we actually explored in our attempt to harmonize data across sites is deep learning models, particularly StarGAN, which is try to learn the distribution of different scanners and protocols, and try to, when they encounter a new scan, they first map it to a standardized distribution, which is shown here on the right, and then you do any analysis. So this way, you have the ability to take scans that look like this at the top, and map them to the style of the scan of the bottom, which is now the middle row. So essentially, you've changed the style in a way that it becomes compatible with others with other studies, so you can actually pull the data together and build more robust machine learning models. So we applied some of these methods to the data before we can get classifiers working well across institutions. And we had a paper back in 2017 where we showed that there's a robust structural MRI signature across at least 3 sites at the time. Now we have, it's over 10 sites or so in the Phenom Consortium that interrogates areas of reduced brain, cortical volumes in a number of regions, including, prefrontal, orbitofrontal, some parietal regions, parieto-temporal regions, and so forth. A lot of emphasis in the perisylvian cortex. And these are some of the regions here that were atrophied in blue, but there were some regions that were increased in red. And I will highlight those because we didn't really anticipate that. Of course, some of them are the ventricles, which we know that their ventricles are relatively enlarged in schizophrenia, but the anterior limb of the internal capsule, the pallidum, some of the deep structures came out as larger. And I'll come back to this later in my talk when I talk about heterogeneity. So as we put all this data together, we decided to apply for a grant at the Brain Initiative and to propose essentially to create brain charts, neuroimaging charts that allow us to put all the data together and leverage it to develop normative distributions of different brain structures or functional networks or diffusion properties, and create things like the one, the plots that you see here, where it shows that the x-axis shows an Alzheimer's signature measure using some, I think in this one is a support vector machine type of, or maybe a random forest type of method, and then the brain age gap method. And the people who are cognitively normal over here, who are gray or green, people who remain stable in longitudinal studies, essentially have negative Alzheimer's signature as expected, and in general, a pretty good brain age scores. But as people start to progress to MCI and Alzheimer's disease, and some of these arrows indicate longitudinal trajectories, you start to see people expressing more and more of this Alzheimer's signature going forward. So other types of brain charts that we've tried to create are shown here. This was from an Alzheimer's and dementia paper where we found that these are isocontours basically of brain age. This is isocontours of amyloid positivity in a population, largely, I think, mostly coming from ADNI. What this plot shows here is that chronological age in the x-axis against the white matter lesion volume with these isocontours showing the amyloid beta status. It shows essentially that if you have increased load of white matter lesions, you're almost twice as likely to have amyloid in your brain, and that's regardless of age. So this is something we didn't expect because these two are considered to be kind of orthogonal. But it turns out that the presence of amyloid, the presence of lesions in your brain doubles your probability of being amyloid positive. So you start exploring relationships of this sort. So now I'll get into the kind of the main part of what I wanted to talk about, which is how we tease out heterogeneity of biological patterns. And as I said earlier, building a machine learning model that tries to identify disease at an individual level is okay, but and unfortunately most of neuropsychiatric and neurodegenerative diseases as well as cancer are highly heterogeneous, and many of them are it's not even clear that they're a single disease. They might actually be caused by different underlying neurobiological regions. Certainly in cancer, we know that different mutations give rise to different radiologic phenotypes, and that can be picked up by machine learning methods. So we put some effort on trying to understand this heterogeneity and build methods that kind of dissect the heterogeneity. And instead of looking for a single imaging signature of, say, a disease of interest, we find multiple imaging signatures if there is evidence in the data that there is such heterogeneity. Of course, one could apply a clustering method and subtype individuals into different subtypes, but that is prone to biases from a variety of factors that influence a brain structure or brain function that are unrelated to disease. So, demographics is the first obvious factor that could influence variability. Of course, before you apply a clustering method, you can correct for age and sex, which is the obvious thing to do. However, there might be other genetic and demographic and other factors that cause variability in the brain that has nothing to do with the disease process that you're interested in. So to address this, we built a weakly supervised learning methods using deep learning generative models. That was a paper in Nature Communications a couple of years ago that Zizang, one of my applied math students, developed. And really the idea here is very simple. It's pretty much what people have been doing in computer vision and for some time now with the generative models. So you can take a Vermeer image and morph it to a different style or to a related style using these generative models. And they are pretty good at doing that, whether you're using GANs or diffusion models or anything else. You can learn essentially transformations from one style to another and therefore generate these types of images. We kind of do the same thing here, but we learn transformations from what is on the left, which is an MRI of a healthy individual, to an MRI of an individual with schizophrenia in this particular case. So by training these networks to find what transformation is needed to be applied to this image in order to generate this image, we actually gain insights into what the disease does to brain structure in this particular case. So I don't have the time to go through all the details of the methodology, but essentially the method says that I have a distribution for healthy controls that is by a reference population. It doesn't have to be healthy controls versus patients. It could be, for example, if you're trying to understand the effects of aging, you can use a young and healthy population as your reference and everybody else as your target. But let's assume that this is a healthy control population, and this is a patient population of interest, say people with schizophrenia. You're trying to find a number of transformations that map this distribution to this distribution here. And these transformations, presumably, if there are multiple such transformations, there are potentially multiple subtypes of disease or multiple mechanisms by which schizophrenia affects the brain. And this is kind of captured by the latent space here by variables Z that at a simple level captures, say, subtypes of disease. Say that there are two subtypes of neuroanatomical subtypes of schizophrenia. We can capture them by having these variables in the latent space that learn different transformations depending on which subtype you belong to. There's a bunch of other tricks here that one has to play. With this, we used GANs here, so you have to, as you generate this generated MRIs of people with schizophrenia, people whose brains look like those of patients with schizophrenia, you have to compare them with real data so that you make sure that the discriminator here doesn't see any difference. That's how the GANs work. And then there's an inverse mapping G here that says if I give you an MRI, you should be able to tell me from which subtype it came from. Otherwise, your transformations might not be believable or might not be interpretable, actually. There are other constraints there, such as continuity, monotonicity, and other things that were important for making these GANs work well, especially in the continuous case, not in the dichotomous case of classification into disease or not. But when you want to capture different dimensions of brain chains, the degree to which a pattern is expressed in an individual, it's a continuous problem that becomes a lot more difficult. So we solved these problems, and we have been applying these methods to data from different studies. This is one of the studies that we had in 2020, and to our surprise at the time, we found out that even though 2/3 of the schizophrenia patients seem to belong to one subtype and they express the same type of brain changes that have been published in the literature amply with orbitofrontal, prefrontal, perisylvian, and some other areas of reduced brain volumes. And this 63% of the patients was the majority, but there was about 1/3 of them that looked different. They seemed to have completely normal brain structure and increased deep structures. And this was persistent with leave one site cross-validation. This was from multiple sites, and I don't remember how many. I think it was probably 7 sites or so at the time. So leaving one site out didn't change the results. We adjusted for CPZ equivalent for medication, just in case this was medication effects. We did split samples. We did, men and women separately, and it was a pattern that was pretty clear. And it turns out also that if you plot the brain volume of, for example, of the first subtype as a function of age, these were the people who had the degenerative type of measurements. In other words, their brain volumes were going down as a function of age, as opposed to people who had the second type, whose brain volume seemed to be not changing with age. So then we went on to, we were intrigued by this, and we tried to understand these subtypes a little more. So we went to the UK Biobank and the Philadelphia Neurodevelopmental Cohort. In the PNC, the Philadelphia cohort of adolescents, basically, Raquel Gore and her group had defined a variable called psychosis spectrum, which is a very broadly defined variable. This was a community-based sampling, but some people, it turns out that almost half had some symptoms, one or another, that put them into this very broad vague category, which the vast majority of these teenagers will never develop neuropsychiatric disorders. But there was some reason for them to go into some very broad spectrum. And we applied this schizophrenia-derived model from the Phenom Consortium. We applied them to these teenagers, basically, and we restricted it to only AIDS comparable populations, so it was only ages 16 and up. And we found out essentially that this bar plot here shows the gist of it, that if you look at the healthy controls in Phenom, which is in gray, and if you look at the PNC people who were typically developing, and even the same thing, UK Biobank, actually, if you go, they had low scores of expression of this S1 anatomical signature, which is basically this type of, reduced brain volumes in this particular pattern that we have seen in schizophrenia. But people with, of course, with schizophrenia in the Phenom, they had increased signature as expected by construction, basically. But people with psychosis spectrum in the PNC, where the green ones, were in between. So these are teenagers who have, most of them do not advance to do not progress to any clinically defined illness, because it was, like I said, about half of the population. It was a very broadly defined spectrum, but they do on average tend to have increased scores in S1. So there's something happening in the brain that is not necessarily clinically significant, but it was also evident in the cognitive profile of these individuals. People with S1 signature at the PNC had reduced cognition. The same thing was the case at the UK Biobank. The trail making test is opposite. So basically this is reduced cognition over here. So people with S1 signature had reduced cognition, and people with S2 signature actually had increased cognition, cognitive performance. So, and what was perhaps the more tantalizing result there, that's from the American Journal of Psychiatry paper that we had. It was that if you go to the UK Biobank, and again, you look at people who don't have schizophrenia, we excluded anybody who had a diagnosis. There are very few people who have a diagnosis of schizophrenia. And we looked at the presence of this imaging signature by applying the machine learning model there. We found that the people who expressed S1 had significantly higher polygenic risk score compared to the controls. And that indicates that these are people who have absolutely no clinical diagnosis, but if they're expressing this pattern that we have extracted from schizophrenia patients, they also have increased polygenic risk score, they have reduced cognition, and so forth. So it's very interesting, and again, it emphasizes again something that I think most people would expect, that these clinical phenotypes are just the tips of the iceberg, and that there's a lot more happening underneath, including things that are detectable in cognitively normal individuals that do not necessarily translate to clinical phenotypes. And that's, of course, these are potentially biomarkers of vulnerability or early detection, or whichever way you want to see it. But it's interesting to see that this heterogeneity analysis methods start to pick up some of these characteristics that that would not be immediately appreciable otherwise. We've applied these methods to data from Alzheimer's disease. There's more data here we've been playing with. These particular results are from, I believe, primarily from ADNI or ADNI and BLSA. And this was a paper that was in Nature Communications in 2022 or so. And what we found is that, again, there were two predominant dimensions. Here we used a continuous version of this clustering method. So we teased out dimensions, neuroanatomical dimensions associated with Alzheimer's disease or MCI. So there are two primary dimensions here. And one of them is associated with, very limbic, pre-hippocampal and limbic temporal lobe atrophy. And the other one is associated with more diffuse atrophy. And then, of course, there are people in between, and you can plot people in a two-dimensional plot here and have a continuous measure of expression or presence of each of these signatures. So if you put these two numbers here in a predictor, and if you put tau amyloid, ADAS-Cog, and ApoE4 genotype, you have you can predict with very high accuracy which people are going to remain stable over a long period, and which people with mild cognitive impairment will remain stable, and which people will actually progress rapidly to dementia. And these are 95% confidence intervals. So this is really very strong predictor. And this can potentially help in clinical trials, stratifying people to the right trials and treatments, evaluating of treatment effects. If you have a better expectation of what the person is expected to do, then you're more prepared to see if there is a treatment effect, a subtle treatment effect, and of course for patient management. So again, this emphasizes that there's heterogeneity, both within the brain structure, but also across biomarkers that we can explore and leverage to be able to build predictive tools that are much more precise than the estimates we would get if someone just showed up in the clinic with MCI and trying to predict whether or not they will progress. What was even more interesting, I think, this is a paper that Junha Wen, who was a former postdoc of mine, who now has joined the USC team with Artoga and the rest of the team. He took these two dimensions that I was showing earlier, these two dimensions that we found from ADNI, from MCI and AD individuals, and he applied them to UK Biobank and other studies, which are focused primarily on preclinical stages. So he wanted to see if these patterns are detectable, of course, at a more subtle level, but if they're detectable in individuals who are younger and they don't have cognitive decline. And if so, what are they associated with? So to our surprise a little bit at the time, we found out that to our positive surprise, yes, you can detect the presence of these signals, although they are much more subtle, less pronounced. So you can detect these types of brain atrophy in these individuals, but they're not associated with ApoE4. They're not associated with amyloid or tau in studies for which we had amyloid and tau. But they were associated with other factors, with genetics. This came from UK Biobank, with genetics of hormonal function for one dimension, and cardiometabolic risk factors and immune function for the other one. So that, of course, suggests that perhaps some of the earliest events that triggered these pathologic neurodegenerative patterns that we see later progress in Alzheimer's disease, maybe some of the early events are not related to the usual suspects of AD, but they might be something else that lights the fire, whether that's related to the hormonal system or to the immune system or to cardiometabolic factors, and then its progression is mediated by ApoE4, amyloid, and tau, because that's what he found in longitudinal studies afterwards, that even though the initial presentation of these patterns of atrophy was not related with ApoE4 amyloid, their progression in longitudinal studies was associated. So it's very interesting. Of course, it needs much more work to be studied in depth, but it's very intriguing. We also look at at aging individuals, not just people with MCI or AD, but people, a much broader population of aging individuals by essentially applying the same methods to a reference group being the young and healthy people, or the younger and healthy people, and then the target group being everybody else. And we expect a lot more heterogeneity there, so it's a good testing ground to see what's happening. And we found out, this was a paper that just came out in JAMA Psychiatry, that there's essentially, these methods allow us to pick out a trajectory from resilient to typical aging. And resilient aging is associated with genetics that are protective for cardiovascular disease. People with resilient aging also have low levels of cardiometabolic risk factors. They have brain age that is approximately 5 years younger than their chronological age. And then after some point, there seems to be a progression, a divergence into two different types of patterns. One of them we call vascular aging, but actually it's combination of both vascular and AD factors. So these people had patterns that had more white matter lesions, which are, a small vessel ischemic disease for the most part, higher cardiometabolic risk factors, genetic genes that have been associated with white matter hyperintensities. They were enriched in ApoE4, which is a genetic risk factor for AD, of course. They were enriched in amyloid beta, and they had higher progression to MCI and brain age approximately 2 to 3 years older than their chronological age. However, the other path was something we couldn't quite explain, we cannot yet explain. They had the most aggressive patterns of atrophy, but they were not enriched in ApoE4 or amyloid. They were not enriched in the white matter lesions, but they had higher incidence of depression. So these people are getting very pronounced atrophy, but not for reasons that could be associated with either cardiovascular or Alzheimer's disease. So again, there are no definitive answers here to anything, but it's very clear that these methods allow us to dissect the heterogeneity of aging, of brain aging in this particular case, and start to understand what the different trajectories of brain aging relate to in terms of genetics, progression, clinical progression, pathologic processes, and so forth. So because we kind of like these approaches, we kind of further build on them. And Ji-Chang had another second follow-up paper in Nature Communications last year, where he says, okay, well, I'm working in this with these latent variables that capture the different types of transformations that my GANs, that my generative model needs to apply in order to model disease effects. How about if I add input from genetics in this latent space to kind of require that these transformations are also genetically explained? And the reason for that, of course, is that this is a way to arrive at endophenotypes of disease, so patterns of brain change caused by disease, but also explained by genetics. So he basically took the same approach as before. He built on that, except that in the latent space, which is again denoted by Z, this is a bunch of latent variables that before they were just express the subtype of disease that we're looking at, which influences the transformation that the generative model will perform. Here we have three families of variables. Z2 are the ones that are explaining only phenotypic variability, so brain chains. Z3 are explaining only genetic variability, and then Z1 is explaining both genetic and phenotypic variability. And Z1 is the one that you want to look at if you're looking for endophenotypes, basically, of the disease. So his networks were exactly as before, which is essentially, this is what I had shown before. It's a GAN It's part of a GAN architecture that that generates brains that cannot be distinguished from those of patients. And of course, the input is brains of healthy controls. And then the genetics is using variational inference that they're linked with brain phenotypes in the latent space here, and they try to express genetics. Again, in terms of the results, again, looking at ADNI, we found essentially 4 endophenotypes. One is essentially associated with very normal brain structure. This is gray matter density maps and white matter density maps. The second one was associated with very strong hippocampal and medial temporal lobe atrophy, and also perihippocampal white matter atrophy. The third one was more advanced, and the fourth one had non-AD-like, non-specific atrophy, both in gray and white matter. White matter was actually not atrophied. It was only in this area here. What was interesting is that if you look at the genetics of these, there are significant differences in the effective allele frequencies of the different subtypes, which are in different colors here. A1, A2, A3, and A4 are different subtypes, and multiple targets came out as being significantly different between these subtypes. And this is, of course, what further indicates that these anatomic differences might be explained by these genes. He also looked at plasma biomarkers and CSF biomarkers, and a number of them were significantly different across subtypes. And the same thing with CSF, with amyloid and tau, as well as clinical scores. For example, A3 here had the highest level of amyloid, and A1 had the lowest level of amyloid as expected. A4, even though it had quite a bit of atrophy, was kind of in between, which is also expected because it seems to be the non-typical AD type of atrophy, and that is further confirmed by amyloid and by actually genetics and so forth. A number of plasma biomarkers came out to be significantly different among subtypes. Many of them are related to immune and cardiovascular system risk factors, and that's also not unexpected. So again, there is no definitive conclusion here, other than what we call aging is and cognitive decline is associated with different trajectories, and the trajectories might have different genetics, different cognitive phenotypes, different amyloid beta and other factors, different immune system and cardiometabolic risk factors and so forth. So this process of aging, cognitive decline, and potentially and eventually dementia is highly heterogeneous, and unless we really break it down into a richer set of dimensions, then we will not be able to fully understand what exactly is going on. Finally, and I would like to end a bit early so that I can leave time for discussion. So finally, we've applied some of these methods to data from ABIDE, again, which is from an autism spectrum disorder population. This is not my area of expertise, so I have to admit that we were just curious, and we were playing with this data, but we got some interesting results. And this is a paper that was published in JAMA Psychiatry a couple of years ago. And so we found that in autism there was tremendous heterogeneity as expected. And if you were to compare ASD with controls, you would find no significant differences basically in brain structure. But if you were to apply this heterogeneity analysis methods, you will essentially find 3 different dimensions or subtypes. A1 had significant brain loss or reduced brain volumes in a number of cortical and subcortical areas. It looked so A2 looked more like one of the infrequent subtype of schizophrenia. So it had the otherwise preserved brain but increased deep structures. And then A3 was the increased brain volumes across the board, especially in the cortical area, perisylvian area, and so forth. And they had different genetic loci that came out of the analysis. And what is essentially interesting is that the A1 expression and the S1 expression was S1 and S2 were the signatures of schizophrenia, and A1 and A2 were correlated between them, especially yeah, both of them actually. But it's kind of interesting that A2 and S2, this increased basal ganglia signature, was correlated in the population. What was also interesting is that only A3 was correlated with ADOS. So that indicates again that and A3 was also the only one that showed significant correlation with medications. So the conclusion was that A3 was the more typical ASD signature, and A1 and A2. This came actually from data in UK Biobank, so it's not necessarily directly comparable because of an age difference, but in the UK Biobank, A1 and A2 were associated with diabetes, cholesterol, higher brain age, and higher PANSS scores in the Phenom consortium, actually in the schizophrenia in the cohort with schizophrenia patients, whereas A2 was more associated with white matter lesions, UK Biobank. Again, there's a lot of heterogeneity in the number of neuropsychiatric and neurodegenerative diseases, and we need to. I think there's the great opportunity to use these machine learning methods not just as a classifying to yes or no, or this is a patient or this is a control, but to be able to tease out dimensions that express different patterns that are of interest. And I spoke primarily of structural imaging patterns, but we're also applying these methods to resting state connectivity and other types of measures. So it's interesting to extract dimensions that tease out patterns that might allow us to obtain more rich representations of what's happening in the brain with disease, rather than try to come up with a single classifier. And that will enable us to define diseases according to biomarkers, rather than to coarser clinical phenotypes, and potentially stratify people to appropriate treatments. Thank you so much. I think that's all I had to say, and feel free to ask any questions now.
Thanks so much. That was great. So yeah, please feel free to unmute yourself and ask questions.
There was something in the chat that I think I saw at some point.
Yeah, I didn't see.
Yeah, I don't know what I don't see the chat.
There are a couple of questions on the chat.
Great.
Should I read them? Okay. So David asking, the first question is from David. What would you say has been the key factor in development of all these methods to find a meaningful subtype?
What has been the key
factor in the development of all these methods to find a meaningful biotype?
I don't know. I wouldn't say that there's a key factor. I mean, there's different people apply different methods. Like I said, one of the things that we're doing differently is that these are what we call weakly supervised methods. So in other words, okay, so the clustering methods, there's a number of clustering methods. They take a population, you apply a clustering method, and you find 3 different clusters. Now, but that doesn't necessarily mean that these clusters indicate different disease subtypes. You might have clustered your data according to demographics, as I said earlier, which you can correct for upfront, but there might be other genetic and environmental factors that have caused this variability that is completely unrelated to disease. So there's a number of clustering methods that are using different criteria, but they are all subject to this vulnerability that you might be finding variability that is not disease related. So what's unique in our approach is actually that we're not clustering brain MRIs, we're clustering transformations from healthy controls to patients. So we observe what it takes to morph a healthy MRI scan to that of a patient, and then we apply clustering essentially in these. So that's a weakly supervised kind of method, because it says these are patients and these are controls, but I don't know which subtype they belong to. So you basically looking at and together with a generative model, you're building up a disease model on which you're applying your clustering. So I think this is fundamentally different philosophy from some other clustering methods out there. So I think that does make a difference. There's other methods for subtyping. There's a method called sustain that has been developed by Alexandra Young. That uses an event-related model, so it uses a different criterion. And it assumes that if you enter a disease subtype, you keep going along it with increasing kind of intensity. So it has some assumptions, but if that's the case, and you don't allow for overlap between the two, then you know that method might give you essentially distinct trajectories that people follow. So there's many different methods out there. It kind of depends on what you believe more in and what fits your data and the disease or pathology you're trying to cluster.
And would you mind to give a comment on why these transforms do not carry the same comment or critique that you raised about the clustering? Why they are not sensitive to the same properties?
Oh, that's a very good question. Well, they could, and they are somewhat vulnerable as well, but they're less because essentially whatever variability is, of course, if the disease effects are related to say demographics, then the transformations will. But this is something that is not confounding. You want to know if the disease has a different pattern in men or women. What you don't want to know is if all the other factors that are different between men and women are causing you to cluster them according to something that is not disease related. So just the very principle of taking the entire variability of a healthy control population or a reference population and mapping it to the patients basically focuses on what is different between patients and controls, and that removes whatever is not disease related. But like you said, if truly these transformations depend on, say, demographics or other factors, that will influence the clustering, and that might be a good thing, because that is a good thing, because that's information that you want to preserve as long as it relates to the transformation from healthy controls to patients.
Cyrus is asking, when you're registering ventricle in all population, how does it affect future analysis if one registers with few parameters, create a subject that have a larger ventricle than MNI versus non-parametric registration? So I guess the main question.
Yeah, I mean, that's, registration with big ventricles has, been a problem. I mean, a lot of these methods have now been worked out, and they can deal with very huge ventricles. The methods we use right now, actually, they used to be based on these, diffeomorphic transformations, and which, if you set the parameters properly, they can actually capture a lot of deformations. But now we have used those to train deep learning models that do the segmentation in seconds. We went through a series of identification of problematic cases with big ventricles. We retrained the models and so forth, and now they're doing pretty well in general. So we don't see any problem, except for very extreme cases with very huge ventricles, and that's always a problem. And it was a problem, especially with these deformable registration methods. Now with deep learning, you can actually teach it to correct for that. But it's certainly an area of concern. Yeah.
So the next question is from Alex Lee. The question is, you mentioned earlier about different aging profile related to depression relative to MCI and AD. But MCI and AD can present with depression symptom. Could you comment on how your model might distinguish between depression related to AD versus other manifestation of the depression in aging?
Well, the method is agnostic to depression, of course. So the question is, if if the people who had the arm that was unrelated to amyloid and cardiovascular factors, if they had increased depression, if they had different types of depression symptoms than the others, or yeah, we haven't looked at this in detail. The depression data came from UK Biobank for that particular study, which is not very well characterized, but we have seen that in people with geriatric depression, we had a paper actually on that, they can have quite a bit of heterogeneity in terms of their patterns of brain atrophy, and so potentially the underlying neurobiological causes of this depression might be different, are likely to be different.
And anyone who want to ask their question, just let me know to unmute. The next question is from Yuping. The question is the deep learning model interpretable for AD subtypes, or how do you identify significant genetic signature associated with imaging?
Say again the first part.
So is the deep learning model interpretable for AD subtyping or not? I think that's the question.
Yeah, I guess interpretability is very challenging with all these deep learning models. I mean, the interpretability we have tried to enforce. First of all, it's not clear what is interpretable, what's not. There's no a quantitative definition of what is interpretable. But the way in which we try to encourage interpretable models is by having that inverse function that I showed earlier, the G function, that says that if you have your generator that generates that does a forward generation of the transformation that you need to apply to transform your image, and then if you're given an image from a patient, can you actually estimate what subtype it came from? Generative models can generate images that look realistic, but they don't necessarily. If you go to the latent space, which has the variables that generate an image, an example, you don't necessarily have a structure that is interpretable. As long as the generator can generate valid images or shapes or whatever it is, it's fine. Now, by having an inverse map, you actually say, I not only want to generate valid MRIs, but I want to be able to recognize which subtype they came from. So that inverse consistency enforces the latent space, which decides what belongs to subtype 1 and what belongs to subtype 2. It encourages to find recognizable patterns in the data rather than something random. So that kind of has helped create more, interpretable or more likely to be biologically based patterns of brain atrophy that we see in the data. Of course, the interpretability for us, we just look at the subtypes. We compare them like voxel-based maps, and then we try to see if they make sense or not. I don't have a clear definition of what would be interpretable, and this is certainly an important issue to be addressed, especially if we come up with a definition of interpretability. One could impose it on the architecture of these deep learning models.
And the rest of the question was how you identify significant genetic signature associated with imaging. And Yuping, if you like, I would unmute you for a follow-up question.
I mean, the genetic signatures are basically they're also identified the same way as the imaging signatures, because the genetics and the imaging data meet at the latent space with these variables Z1, Z2, and Z3. So basically, the genetics and the imaging meet in the latent space, in the low dimensionality space, in the bottleneck space, basically. They meet there and they share variables that then help the respective generators generate the data. So that's where the imaging and the genetics data are forced to be mutually informative, if you want. And not unlike methods that Vince and others have used as well in the past, things like canonical correlation analysis that have used these kinds of concepts. With deep learning, basically, and I know that Vince has used a lot of deep learning as well with the same concepts, but in the latent space gives you the opportunity to merge the different data in a way that it's actually to partially merge, if you want, because some of these latent variables can explain only brain variability and some can explain only genetic variability.
As a follow-up question from Cyrus, he's asking about so the question is about the registration ventricles, about registering ventricles in all subject. May you have a reference to a good method in publication? Would you recommend any specific paper or public?
For specifically for ventricular segmentation?
So registering ventricle in all subject. So I guess the question is because we have like a lot of atrophy or deformation. What's the method you recommend?
Yeah, so in the past we've been using this method called Muse, which is based on ants that the diffeomorphic illustration and grams and other. It was a multi-atlas segmentation, multi-warp. So it was using both multiple atlases and multiple warping methods to be able to kind of have a little bit of an ensemble. So the idea of these multi-template methods is that if you have big ventricles, there are some subjects, you don't use a single template, especially a template with small ventricles. You have an ensemble of templates that they all are deformed to the individual of interest, to each subject. And the ones that look more like your subject are the ones that have more of a say, if you want, of a vote. So that's how we had solved them back then. Now, this is old work. This is more than a decade old. When you have multiple templates voting, and then the ones that are more suitable have more of a vote. So that's how you get over this situation with large ventricles. You have an atlas ensemble that has captured the variability essentially. And these atlases came out of a large data set which we clustered. So that it actually, you have about a dozen or so atlases that capture the variability that you want to capture. So you have enough variability in your templates. Otherwise, if you're using a single template, yes, I absolutely agree. It can be very problematic because if you're using a young and healthy brain and you try to work with a 90-year-old demented individuals, it's going to be very challenging.
Thank you. And the last question, I hope I don't mispronounce his name. Mick is asking, first of all, he's thanking for the great talk, which I agree completely. So he's asking, or she, could you share your thought on how medication effects may be reflected by dimension? And how can we account for the tight relationship between disease severity and medication intake? Good question, very good question.
Yeah, that's a good question. And that's the first thing we thought about. And everybody said when they saw the second subtype of schizophrenia, the one with the increased deep structures, because it has been reported that there are, subcortical increases with medication. So we corrected for CPZ equivalence and the relationship was still there. So certainly some of this might be due to medication. There's no magic bullet here. You either explore this relationship afterwards to see if this dimension that you picked up is truly, related to medication, or if you're confident that you want to correct for medication in your input features, you do that and then you extract the dimensions.
Yeah, or ideal, or if you have like a early stage unmedicated sub data set, probably can also check on theirs too. Yeah, thank you. I think there is no other question.
Thanks so much. Yeah, so I guess one last question I had was, so how close are we to a clinical tool?
That's very interesting. Yeah, I mean, that's what we tried to do with NiChart, and also some associated efforts where one of my colleagues, Ilya Nasrallah, a neuroradiologist, who is also a computational scientist, is trying to put this in the clinic. We've created some pipelines here, prototypes that take data from the PACS, and then they run it through AI pipelines. I mean, this is the usual question I get every day. I mean, I've gotten this question yesterday, the day before as well. I was giving, radiology rounds yesterday. I don't know that there's a clear answer. Within institutions, I think prototypes are certainly implemented. There are both cultural, regulatory issues. The technology is running too fast. I mean, 5 years ago, things were so different than they are now. So any effort to take a product through FDA clearance and everything else, it will be obsolete by the time it makes it out there. It's a very rapidly changing technology. There are some tools. I was just talking with Paul actually yesterday, Paul Thompson, in that AI for AD meeting, and then he mentioned this tool called BrainC, which does exactly what I was showing before, predicting which people with MCI will actually progress. So there are some tools that have made it to the clinical practice. I think whether or not they will be adopted, they will change the practice. It's to a large extent cultural. How do you get people to stop what they're doing and then and change things? But some of it is changing. And so for example, now Ilya that I mentioned before, I don't have a slide with his, but he's creating clinical reports that are not just the usual neuroradiology reports of reading brain MRI scans, but they also have specific Z-scores for different brain structures. You have your Alzheimer's signature, if it's positive or not, your brain age. So it's the stuff that we've been doing in the lab for a while. Now it populates standard clinical reports. So that's very exciting. To get it to every clinic's, every radiologist's hands, you have to run it through FDA approval, which is the usual, regulatory hurdles and everything. But it's a good question. I think with the one thing that we've seen recently, of course, in the AI world with OpenAI and LLMs is that sometimes it looks like a technology is not that it takes forever to be adopted. As you know, AI tools have been in the development for years and years. And all of a sudden, when OpenAI made available the LLM model, everybody started to use it with ChatGPT, and everybody now talks about deep learning and AI in all fields. So sometimes these changes are, quantum changes that they take a while to happen, but when they happen rapidly. So I think that something like that will happen. It will happen in radiology as well as other clinical specialties.
Yeah.